Hello, everybody. This is Dr. Phil from Med Pathway, your online MCAT Prep Testing Center developed by med school professors. Today, we're going to talk about viruses. A lot of viruses in the news these days. So let's take a look. Here's bacteriophage. This is the simplest virus of all. So basically what you have is a capsid structure. It's a protein coat that surrounds the genome, which could be DNA, RNA, single-strand DNA, single-stranded RNA. Then you have over here a collar and a sheath. This is like its body, its abdomen, so to speak. And then you have these tail fibers here. These tail fibers serve to actually adhere to the host cell. It usually binds to specific membrane proteins that allow the virus to inject its DNA or to even enter the cell. So if you extracted this virus in phenol, what you would do is you get an aqueous and a nonpolar layer. And then what you would recognize is that the genome, either RNA or DNA, is going to be in the actual aqueous layer. And a lot of the proteins will be in the nonpolar layer. So this is how people actually measure viruses. So what they do is they infect cells and they plate them out on agar plates. And these plates have a bunch of host bacteria in there. What you see are these holes that are called plaques. And they're slow growing areas, or in fact, they might even be lyse colonies. Not all viruses are going to kill or lyse their cell. And what you can do is actually quantitate this and count them and get what's called a titer by the number of plaque forming units. So you may see some stuff about quantitation and titer of viruses. But viral life cycles is what we're going to focus on right now. It's pretty important to understand these. And so we'll take a look. So what we have is a bacteriophage binding to a host cell membrane, injecting its chromosome. Well, it's not really a chromosome. It's actually its viral genome into the host through like a syringe-like mechanism. And here over here in orange is the host chromosome. And then afterwards, the virus is done. It's like a suicide injection of its genome. And then what happens is that in the lytic cycle, you degrade the host chromosomes. So the viral genome is actually expressing proteins that serve to hydrolyze the genome. And then later on, what you do is you make more viral particles, you start to package them, and then they explode out of the cell and you're in what's called the lytic cycle. But there's also the lysogenic cycle. This is what you see here. So you have the same initial thing where the virus binds, injects its DNA into the host, but this time the actual viral DNA doesn't blow the host up by lysis, but rather what it does is it integrates into the host chromosome known as a prophage. This dormant, silent viral genome is sleeping along and it propagates as the host unwittingly replicates it. But imagine that this host, after you replicate multiple times, imagine the host cell, the bacterium gets into danger where there's no nutrients, etc. The virus can sense this and it's time to leave. And so what it does, it actually has SOS mechanisms where it can get out of the lysogenic phase, re-enter the lytic phase over here, and then try to find a new host in which to thrive in. So you have the lytic cycle and the lysogenic cycle. So in the lytic phase, there's a phenomenon known as general transduction. And so what you see here is that the bacteriophage has injected its DNA. It's on step 2, what you see is degradation of the host chromosome, and then you repackage in step 3. But every once in a while, some of these phage, what are they going to do? They're going to actually mistakenly place some of the host chromosome pieces into its capsid as if it were its own genome. And when it goes to infect a new host, what you get is a transfer of not viral genome, but rather host chromosomal DNA. And what happens is that this new host here will have possibly the gene that's came in through the virus plus its own gene. So it could have 2 copies of that gene. And we know that bacteria are typically haploid, but in this case, you can be partial diploid. There's also specialized transduction, and this equates to the lysogenic state. So in this case, we see the phage injecting its DNA into the host. We make the prophage. And then what happens upon an SOS signal, the virus can actually, when it comes out of the host chromosome, it makes a couple sloppy mistakes and actually takes flanking chromosomal DNA from the host and it packages it into its capsid. And this can actually be used to horizontally transfer genetic information to a new bacterium, creating a partial diploid. So there's specialized transduction and general transduction. So, you know, the way we paint the picture is that bacteria are these unwitting hosts that just get knocked out by viruses, but the bacteria have evolved various immune defenses against these viral invaders. And what we have, one of them is called host restriction modification. This is constitutive, it's always active. And so what the host does, it methylates its own DNA. And by having it methylated by EcoWAR1 methylase, for example, there's many types of restriction enzymes. What that does is it becomes resistant to EcoWAR1 endonuclease. And what happens then is by having these enzymes constitutively expressed, any viral invading DNA that's not methylated, it's not host, will be cleaved by EcoWAR1 at these specific sites and ultimately be degraded. And so methylation of the host genome defines self versus non-self. And what you see then are two distinct enzymes, EcoWAR1 and EcoWAR1 methylase, that can recognize the exact same DNA sequence, but perform different catalytic functions. And what we call these, the relationship between the two is known as isoschismers. That's a great term. So restriction enzymes, their discovery, their purification is the birth of recombinant DNA technology. In fact, in order to splice and dice genes from one organism to another, you have to use restriction enzymes. At least they did in the beginning. And Genentech, a famous biotech company on the West Coast, actually were the first guys to produce human insulin using recombinant DNA technology. But so what you see is there's also CRISPR CAS. This is adaptive immunity, and adaptive immunity is when the host remembers the invading event. We're going to look at immunology with B and T cells a little later in one of our workshops, but what we have now is bacterial immunity. So what's CRISPR Cas? There's been a lot of talk about it. CRISPR means clustered, regularly interspaced, short palindromic sequences. That is a mouthful. And here's how it works. So what happens is that you have an invading virus inject its DNA into a host, and there's a bunch of Cas proteins. We're not going to differentiate between all the different types of Cas proteins, but this Cas protein here will guide this double-stranded DNA in and integrate it by recombination into the host in a place which is called the CRISPR array. These are repeating sequence elements, and people had noticed them as far back as the 1980s. They were sequencing bacterial genomes and saw a lot of repeating units. And when you integrate the viral DNA from the invader into the CRISPR array host, it's called acquisition. So the host is acquired foreign DNA, purposely inserts it into a locus, and then you can express it, make pre-CRISPR RNAs, like a messenger RNA, so to speak, but it doesn't translate into protein, so it's a sterile transcript. And then what happens is that you can cleave them. And then the second time, if there's another viral invader injecting its DNA, what will happen is that this CRISPR RNA, guided by the Cas proteins, will actually recognize it through this RNA sequence and cleave and cut the DNA and hopefully eliminate the virus. Now, people have observed this and have used it to actually specifically edit the human genome with respect to the system, but this is the actual biological adaptive immunological method. And so the Nobel Prize in Medicine was given in the 70s to the gentleman who figured out the host restriction modification system. And recently in October of 2020, Charpentier and Doudna were awarded the Nobel Prize in Chemistry for CRISPR Cas. So now let's take a look at animal viruses, and what you can probably guess what this is. This is the coronavirus, and you see the telltale spike protein. And so the Baltimore classification of animal viruses states there are 7 different types of viruses. And what you can see is that there's all types. There's double-strand DNA, single-strand DNA, double-strand RNA, single-strand RNA. But by hook or by crook, what's going to happen is that the virus is going to make a messenger RNA and a bunch of proteins that are going to make more viral particles. We're going to take a look at a couple. We're going to look at coronaviruses, as well as we're going to look at the retroviruses. And if you take a look at class 6, the retroviruses, you see a positive single-stranded RNA being converted into a DNA RNA hybrid, making a double-stranded DNA ultimately to an RNA. The positive single-stranded RNA of retroviruses like HIV is shared with coronaviruses because they're also plus single-strand RNA, and the plus means that you're basically like a messenger RNA. Once you're a plus RNA inside of a cell, the ribosome can jump on it and translate. The difference between HIV and coronaviruses is that HIV has a DNA double-stranded intermediate, but the coronavirus has no intermediate that's DNA. And so what this says is that coronaviruses can only be in the lytic phase. They can never be lysogenic because they can't find their way into the chromosome as a DNA entity where they can go to sleep. But HIV is different. And so what you should remember with respect to retroviruses is that the Crick central dogma of DNA, RNA to protein was violated by the discovery of Baltimore and Temin of an enzyme called reverse transcriptase. You see this on the right. We've talked about this before. You should appreciate that what it can do is use 3 different enzymatic functions to convert single-stranded RNA into DNA double-stranded DNA, that can integrate into the genome. And what we also know is that reverse transcriptase is an enzyme used in laboratories to actually do real-time PCR to measure messenger RNA. And so here's the structure of HIV. Here's the pattern of the retroviruses in class 6. So what you see is actually a pretty interesting genome. You first notice it as a lipid envelope, and it kind of reminds you of a typical eukaryotic cell membrane because it's endowed with all kinds of receptors that are derived from its host. Here's major histocompatibility complex. We'll talk about more of this when we see immunology, several workshops down the road. Here's GP120, which is actually a receptor that HIV uses to get inside of the cell. And you should appreciate that HIV only infects CD4 positive T cells using things like GP120 or even CCR5 as the way to get into the cell. Here's the genome. There's 2 single-stranded positive RNAs. You have a molecule of reverse transcriptase, along with other host proteins and the GAG protein from the virus that are all used to make more viruses. And so what you see happen then is that the HIV will recognize a GP120 or some other receptor like CCR5, inject its DNA. Actually, it fuses with the membrane. The DNA is, it's not even DNA, it's RNA. The RNA is turned into the RNA DNA hybrid, and then the double-stranded DNA, this is what I was trying to say, is going to be integrated into the genome, and there it could be lysogenic, and then it can be activated, and then it can translate, make more viral particles, which could leave the cell, and when it leaves the cell, it takes its membrane with it. So this raises an interesting question about PCR and Western blotting testing for HIV. So if you have a PCR test, you can detect the lysogenic state. If you just do a Western blot, what you're measuring from somebody's blood sample is the actual proteins from the viral coat in the serum. So what you can do is a positive Western blot means you're actively have active infections, but a negative Western blot means that you may be lysogenic or you may have no HIV infection at all. And so that's why PCR is a great therapeutic tool to see if you're basically going to be have HIV or not. So people have come up with antiviral chain terminators. Here's what one you see here. This is basically a nucleotide chain. It could be DNA or RNA. If it's H, it's DNA. If there's an OH there, it's RNA. And so what happens then in a typical polymerizing chain, the 3' hydroxyl group attacks in an SN2 fashion this alpha phosphate. Pyrophosphate leaves, and what you do is you make a new phosphodiester bond. But the next time the polymerase comes, if you have an H there, if you chain terminate with no 3' nucleophile, then you can't replicate. This idea of chain terminators is really popular, and Fred Sanger used it to create DNA sequencing. And so the initial HIV drugs use this idea. So azithromycin is the first one. What you see here is azithromycin does not have a 3' hydroxyl group, but rather what it has is this tricked out azido group that probably was designed for solubility, transport, toxicity, etc. And AZT is the first FDA approved HIV drug. And I'm not sure if you guys have seen Dallas Buyers Club with Matthew McConaughey. I highly recommend it. It's a true story about Ron the rodeo clown in Dallas who had HIV, couldn't get access to AZT. It was kind of new at the time. And so he comes up with all kinds of wild schemes to get his AZT. Let's switch gears from the retroviruses and look at positive single-stranded RNA viruses. I'm going to focus on coronavirus on the right, but we're going to do a workshop passage on the biochemistry of poliovirus, which is a pretty interesting creature. So here's coronavirus. Now, there's lots of coronaviruses. There's 3 zoonotic coronaviruses that have infected the human population since the year 2000. So zoonotic means it's transferred from some sort of organism like a bat, which are big reservoirs for coronaviruses, or something else that comes to the human population. SARS came out in 2004, MERS came out in the Middle East in 2009, and SARS-CoV-2, which causes COVID-19 disease, is believed to have originated in China sometime around November, December of 2019. That's still up in the air. And so coronavirus comes in, and it recognizes a receptor called ACE2. People know ACE2 is a receptor, but people are looking if there's other receptors as well. And it uses its famous spike protein to interact with ACE2. It gains access, and basically it injects its positive single-stranded RNA into the host. And we know that is like a messenger RNA. It's got a poly A tail, it's got a 5' cap. The host ribosomes immediately see it, start unwittingly translate viral proteins. And one of those proteins will be an RNA-dependent RNA polymerase, which makes the minus strand, and then that serves as a template to make more positive strands. We have other proteins like spike proteins and other envelope proteins going through the secretory apparatus, getting glycosylated, etc. And then at the end of the day, what you do is you package a new virion, and then you exocytose and you're on your way. But you should recognize, since there's no DNA intermediate in coronavirus, it's always going to be lytic. So how do people study things like spike protein in coronavirus? Well, one thing they use is a system called a pseudovirus. And the way you do this is you take plasmids which encode viral proteins. And this plasmid can encode an envelope protein from example, this could be from the coronavirus and the spike protein, but the packaging plasmid and the transfer plasmid encode viral proteins that are derived from a non-coronavirus species. And what you make is some chimeric Frankenstein pseudovirus that expresses the glycoprotein, the spike glycoprotein of coronavirus. And what you can do is you can isolate these things and you use them to transduce another cell line over here that has ACE2 receptors, and you can study how the coronavirus spike protein interacts with ACE2 and gains entrance inside of the cell. By definition, the pseudovirus is incapable of replicating itself, and that makes the system simplify because you're only studying entry. So pseudoviruses is a very common experimental procedure to focus on studying viral entry. So remdesivir is to AZT and AIDS, and remdesivir is to what coronavirus is. So AZT was the first HIV drug. Remdesivir is the first coronavirus antiviral approved by the FDA. And so what you see here is something that looks like a purine. Here's a ribose ring. And it turns out that this is actually a chain terminator. Now, its results with coronavirus have been controversial. It appears to work, especially in late stages of the disease, but it was originally used for the 2015 Ebola outbreak in West Africa. So there's been a lot of talk about coronavirus testing. So we're going to take a look at it right now with respect to PCR tests and the Western blot. So we know that you can take a swab from like the nasal area or even saliva, and if you have coronavirus there, it'll be a positive stranded RNA that you can convert into a DNA double strand that can be used for PCR analysis with primers that are specific for the coronavirus. And we talked about real-time PCR before. It's a quantitative method. And it turns out that the way it's quantitated is that the polymerase can actually produce fluorescence. And if you achieve a certain amount of fluorescence, a threshold level, due to the number of PCR cycles or amplifications, you can detect it. Now, we talked about how the number of cycles that was required to reach threshold was inversely proportional to the amount of initial input DNA. That's a really important thing to recognize about real-time PCR. So that means that somebody whose threshold was achieved after 20 cycles has far more viral load than somebody whose threshold was achieved at 40 cycles. Now, this is translating clinically into a big discussion about what is the appropriate number of cycles that people should use. Most clinical labs are using 40 or 37. And so when you get your coronavirus PCR test back, all it says is positive or negative. If you're positive, it doesn't tell you that you achieved positive through a threshold fluorescence of 20 cycles or 40 cycles. It's possible that 40 cycles is such a small amount of viral load to begin with that it may not actually be a clinical concern. That's a huge discussion right now. So after the coronavirus has gotten into you, what's going to happen is that hopefully you're going to generate some kind of immune response. And this is how it works. So on day 0, you're exposed, and about a week later or so, you start making IgM. And IgM is low affinity, and it's on the surface of B cells. It's not secreted. But the second exposure is actually going to cause some memory B cells to actually produce IgG, which is actually soluble and more higher affinity. And so when people talk about the antibody test, what you have to really distinguish is whether or not you're IgM plus or IgG plus or both. It's a pretty important concept. And as we do the immunology stuff, we'll take a look at that further. So I imagine that viruses and coronavirus and microbiology should be super high yield for the MCAT, given the havoc that these things are creating on the world. And so what we're going to do now is we're going to do a couple of really challenging workshops on poliovirus as well as coronavirus. And so I hope that everyone comes and sees it.